ICA. Let's start again. So now we've heard a little bit about dimensionality reduction. We heard now cell type identification. We went a little bit now on to the intermediate cell types between and transitional states between one another. But now we're going a little bit more into how you can define a trajectory and use that to compute, for example, differentially expressed genes or how one cell transitions into another. And these type of analyses are called trajectory inference analysis. I mean, there are many names also. So you'll talk a little bit about pseudotime, or some people call pseudotime analysis. There are many variants of that. So this is a presentation that I also got from Åsa Björklund. And today the exercises also, we got the help from Julius Gillette. Actually, he created the exercise. So yeah. Yes. So having the pipeline that we have been working so far. So we have the data that is already normalized. We perform then PCA or ICA. And then we do another type of dimensionality reduction method, either TSNE, UMAP, or diffusion maps, which I'll talk a little bit today. ICA I'll also mention briefly. And then you go for clustering, as you have done. And then you want to get to the trajectory to do differential expression and find which genes are the ones that start the differentiation process of a cell type to another. There are many pathways that you can go to the trajectory. So either, for example, you do PCA clustering, and then from there you can also do the trajectory from any of those steps. So there are several different methods out there, and I'll just briefly mention a couple of ones and give you the theoretical background, how you can understand them, or how can you group them all together. So let's understand a little bit, or differentiate what is experimental time compared to the trajectory time, or let's say a developmental time here. And that's what we would call the pseudo time. So it's a time where the cells are ordered by differentiation process. So here, in this case, developmental time. And let's imagine that you have cells in vitro that you put any stimuli to induce their differentiation to another cell state. And then over time, some of them will start completely differentiation, the differentiation path until here. Then you can see they are completely differentiated. And some of them will be somewhere in the middle. Some of them will not differentiate at all. So these processes, even though you have quite homogeneous cell populations, they all will not differentiate exactly at the same rate and at the same time. But one thing that we can notice is that there is, of course, a continuous spectrum here. So there are, from one cell to go to another, you need to have a continuum of cell paths. And this is also reflects, for example, the transcriptional program for activation and differentiation. So they start expressing some genes, and then they transition to another transcriptional state, and until they get to the final point. And it's nice to note that some individual cells will differentiate at different rates, as illustrated here as well. And each cell can be considered as a snapshot of the differentiation process. So if you have a sample, let's say from the bone marrow or from stem cell development, you see that every cell would be a snapshot at that time of differentiation process. So then if you have like a whole continuum spectrum of them, so then you can define a trajectory in this data set. And as I just mentioned, the pseudo-time is basically an abstract unit of measuring developmental time or any other activation process, or regardless of what are you working on. So it's an arbitrary measurement of time or progress on those. Yeah, so the distance between cells, between the start position and the end position. Go away, yes. So should you run trajectory inference in your data set? So you need to have some things in mind before you do that. So are you sure that you have a developmental trajectory in your data set? So let's take an example like bone marrow, which all your immune cells are generated. So you know that you have some projector cells in there. Or if you take, for example, the blood. The blood, you have all mature cells. You don't have the developmental or the cells that originate the cells in the blood in that sample. So if you run trajectory in the blood, you might find that T cells are related to neutrophils, but you don't have any intermediate cells to make sure that that is the right path. But if you have the bone marrow, you know all the cells, and you have all the intermediate states. So then you know that setting a trajectory in the bone marrow is meaningful compared to the blood that you can set a trajectory, of course, but it's not meaningful. It's not biologically relevant. And do you have intermediate states? So that is important, another differentiation between those two samples. And do you believe that you have a branching in your trajectory? In some cases, you might have only a linear trajectory, let's say developmental of, I don't know, B cells to plasma cells. That's quite linear. There's no branching in that, compared to, for example, T cells that differentiate in many subtypes. So these also are things to consider when you're trying to set. So for example, if you try to do branching on the B cells, you can, of course, try to do that. But is that biologically relevant? So keep that in mind, depending on different, yes?
What would you recommend if you don't suspect it's branching, but you suspect it's a linear?
I think I'll come to your question in the end of the presentation. If I don't answer, write it down and say, Paulo, please answer my question. Yes, I'll come to that soon. So just be aware that any data set can be forced into trajectory. So it's usually good practice that you already know that you have some path in there. At least you have biological indications that that is happening. So for example, if you're studying stem cell differentiation into epithelial cell progenitor, so you know already the cell type. So if something is wrong in the trajectory, you can say, yeah, this is wrong. I need to set the parameters so that the trajectory is adjusted to your biological question. And yeah, and another thing that is important is that the gene set that you use, either the highly variable genes or genes that are differentially expressed between your clusters, captures what you expect. If you, for example, have a trajectory to a specific cell type, and the genes that differentiate to that cell type are not included in the list that you want to set a trajectory, that cell type will not be picked because the genes that differentiate to this cell type is not here. So the computer cannot know that if you don't provide the algorithm with the right information. So be aware. All right. So there are many methods. So trajectory analysis started originally around 2014, which is 5 years ago, with Monocle. Then rapidly increased in numbers of methods. Now we have around 60 different methods. So here's the second Monocle 2 that came out. Monocle 3 is coming now here. It's just Monocle was the first one. So then you have also the path of development of these kind of algorithms as well. And many of those algorithms are also quite related to each other. But that's the idea. And here I'm just illustrating a couple of different trajectories that you can set depending on your biological system. So we have cyclic trajectories, either linear trajectories, bifurcation, multiplication if you have many that originate from a single stem cell, or even trees, or completely other different connected graphs that you can have a linear path plus a cyclic plus a divergent. And these are the more complex ones, and I'll come to that later. So here's also just a nice overview of different methods. So overall, many of those things we have already talked about. We have seen about PSNE, PCA. This is also not updated. For example, we don't have UMAP. There are many other things that are not listed in here. But it's a good overview. So for example, different trajectory pipelines or workflows, they use different methods. For example, for manifold learning, let's say dimensionality reduction, PSNE, PCA, or KNN. Here, I'll talk a little bit about ICA and diffusion maps. There are different clustering methods. The graph partitioning would be OK as well. And then trajectory modeling, there are different ways of doing that, but I'll also mention a couple of ones. Yes. So just before going a little bit into the how to build the tree, how to set the trajectories, I just want to mention two other additional dimensionality reduction methods that are quite common, especially if you go, for example, Monocle 1 uses ICA, and that is quite cited in the literature still. So ICA stands for independent component analysis. It's quite relevant, similar to what we have in PCA, but the idea of ICA is to decompose your data rather than aligning them to the highest component that have highest variance. You want to decompose to the sources of variation of your data. So if you think that biologically, what does that would imply is that, for example, you have some true biological signals from your pseudotime. So imagine some cell that are in the start of the differentiation process to a completely developed. You see, for example, maybe some receptor signaling you started activating, cell activation starts, cell proliferation maybe here, and then marker expression. But when you combine all of those, maybe these ones are also, for example, some gene is affected by all those processes. And when you see in the single cell data set, you only see like a mixture of all those processes all together. So some genes are influenced by all those processes, and then you have just a mixture. And what you want with ICA is to decompose your data to get the original signals. So that's what ICA is kind of trying to do. So if you give the same example as I had for PCA before, so PCA, you have only one line here, let's say. And PCA will try to find the highest variance. Let's go back a little bit. The highest variance is in this line, not even though we really want to put a line in between those cells here, right? But the PCA will find the highest variance in your data set. And then the second principal component will be here, 90 degrees to that first principal component. Whereas for ICA, what you want to find is which are the most independent components that you can find in your data. So then that would be, for example, two independent components, one here and another here, by any kind of angle. It doesn't really matter for ICA. But there are two problems with ICA, is that ICA assumes that the signals are independent, which might maybe not be true for single cell. We don't know that yet, but there are other problems as well. And the values of each signal should not be Gaussian distributed, because, so for example, ICA is quite used for sound detection. So for example, if we put 3 microphones or 4 microphones in this room, and we can actually depict who is speaking, even though everyone is kind of speaking at the same time, because we know using ICA, we can decompose the voice of every single person. The same thing goes for instruments. But those values are not Gaussian, let's say. You don't, in most cases, the biology, we have Gaussian distributions. Even for single cell, we cannot assume that they are Gaussian or not Gaussian. But this is just details apart. You don't really need to memorize all of this. So if you give in that example, so let's say that you have this gene, raw gene expression. This is like an example of sound, that the microphone that I was mentioning. This is actually the true biological source, so actually 3 instruments playing. So here you have the frequencies of them going up or down. This is another instrument that is going like this. And if you do ICA on this raw expression, you can actually decompose your sources of variation, or in your data set, independent components, that matches quite well to these, what you call the true biological signals. But the idea of ICA is that then it's also a linear dimensionality reduction method, and can be used to estimate the sources that compose your data, and that would be quite useful. And these sources are assumed to be independent, which is also would be nice for setting trajectories. So then you can identify which processes are coming first and then the second one after. But this might not be true for single cell. So other problems with ICA for single cells specifically, it assumes that the data is non-Gaussian, which might not be true. And each component also has equal importance. Compared to, for example, PCA, that the first principal component is more important than the second and the third and so on. For ICA, everything is equally important. And the other thing is that ICA cannot identify, so since everything is important, I need to specify how many independent components I have in my data. So if I have my data set with thousands of cells, thousands of branches, how do I know how many components I have? Of course, I can try to set up random numbers to say, or try all of different ones. But this is some of the challenges of using ICA nowadays for single cell. That's why we're going now to the other methods that have been developed after this. So this is actually the first method that was developed. Another method that was kind of in between is using diffusion maps. So diffusion maps, let's imagine that you have some cells and they are connected based on the neighborhood that you have in here. So diffusion maps is also a non-linear dimensionality reduction method, and it's based more on probabilities. And it calculates the probability of getting between 2 points, let's say A and B, is defined of the probability between going to that node. So for example, let's imagine 0.1 and 6, and you want to go to that using 2 steps. So then you can go this way and that way. Yeah, that's actually the only way you can get to that in 2 steps. If you want 3 steps, then you can go 1, 2, 3, or 1, 2, and 3. So you can define the area, go to that via this way using 2 steps, or using this way via 3 steps. So the probability of going to 0.1 to 6 using only 2 steps, the probability of going 1 to 2, and the probability of going to 2 to 6, which is this path here. And from 1 to 6 using 3 steps is the probability of going 1 to 4, 4 to 5, 5 to 6, and the probability of going 1 to 5, to 7, and then to 6. So this is kind of what is kind of measuring. So let's imagine now that you have point A and B in the end of extreme of a developmental path, and you have an intermediate point C that it doesn't really matter which one, as long as it's kind of in the same number of steps from A and B. And you have the probability of going A to C and the probability of C to B. That means if the distance between B and C, or in terms of probability, is the same as C and A, that means that those paths are quite connected, well connected. So actually, the diffusion maps will calculate the diffusion distance between A and B as being the difference in probability between those two. If the probability of those two is equal to 0, that means that those 2 points are well connected via some point called C. And then the dimensional, this is just calculate a big matrix still, and then you decompose that matrix using eigenvalue decomposition, which for now, let's imagine that is somewhat similar to what PCA does. So get the highest variance and the lowest variance and so on. So then you can also sort the eigenvectors based on that. Yeah, so this is how diffusion map would look like, and actually you have these in your exercises. So in summary, diffusion maps is also a non-linear method for dimensionality reduction, and the distance between points reflects the probability of going from one point to another. And again, your data must have any connectivity between them, otherwise these also will fail basically in any dimensionality reduction that you want to set trajectories, including diffusion maps. So yeah, sorry, yes.
The exceptional development pseudotime, would that also be applicable for like cancer cells, where you still have some type of development, maybe not in the same way as differentiating, but there is something that is started and it accumulates changes over time. And,
absolutely, yeah.
Branch to other things, but this would be something similar, right?
Yes, exactly, yeah. As long as you know the starting point, then it's much easier to define trajectories as well. Because then you know that you have your path, you already know the biology a little bit behind it, right? And then you can set whatever developmental path it is, maybe cancer, maybe stem cell biology, yeah. Or activation, immune response, it could be anything.
Yeah.
So any questions so far? No? Yes? No? I don't know? Yes. So now we know 2 other methods besides TSNE and UMAP to reduce the dimensions of your data. Now we want to build the relationship between our cells and define where should I start a trajectory and where should I end it. So one of the methods that is commonly also used in many methods is the minimum spanning tree, MST. And that, let's get an example like here. So we have these different points, and you can measure the distance between those points using, for example, Euclidean distance, or using, for example, diffusion maps as the probability of going one to another. And this distance will reflect that probability. So these points will be highly connected than these ones. So you can also put the distance between all those points. So for example, this point is connected by a distance of 9 to this one, and distance of 2 to this one, distance of 6 to that one, and so on. And this black line here is the minimum spanning tree. So what it is, so for a given set of points, we can connect all of them so that we build a tree connecting every single point so that the sum of this tree, all those values here, for example, 8 and 7, are minimum as possible. So we can try different ways of arranging that tree. So for example, this line here, if I, instead of using the 8, I use the 9. So the sum of 9 plus 3 plus 2 plus 2 plus 8 plus 7, 4, 1, and so on will be higher than if I just use this one. So this one is the tree that has actually the minimum score, minimum connectivity, connecting all the points. So you can actually test, for example, you cannot choose this one because this is lower. So between this one and the rest here, this one is lower than that one. So this is actually the only path that you can connect it, and so on. So in this case, it's very important to have more transitional cells because in some cases like this one, the distances are very far apart. So let's imagine that you don't have this one. Now the transition is not from this one to that one, but rather this one to here, to one of those two. So the trajectory will change quite a lot. So it's very important to have many transitional cells between the two end states that you have. And that also will improve the definition of the tree. And the weights can also be distances in ICA space, correlation, a diffusion distance that I just mentioned, a distance within the UMAP dimensionality reduction that we have done. And by definition, a minimum spanning tree does not have cycles. So if you have a cyclic process, let's say in cell proliferation, you cannot use this method just because of this. Yeah, so you can, yeah, exactly. So you cannot use MST for cyclical trajectories. So I'll come just basically to Monocle 1, which is exactly what I just mentioned. So you have your points represented in a multi-dimensional space. You reduce dimensionality, in most cases ICA, but you can also use any other method, UMAP, TSNE, diffusion maps. Build a minimum spanning tree, and then order your cells based on this minimum spanning tree. And then, of course, you assign. I know that this is my stem cell, so I assign this is where I should start. And then we measure the distance between this point and this branch here, and this point to the next branch here. And that will be basically your pseudotime. So the distance between this point to that one is your pseudotime. Following this path here. And then you can also have your labeled cells, and then you can do other things with it. So here is also, for example, using ICA, component 1 and 2, and you have 3 different end points. And then you decide, this is where I should end. This is one of my other ends. The minimum spanning tree will just try to find the minimum path that connects every single point in your data set. Yes?
What if you get not one minimum tree, but several possibilities?
Yeah, so the minimum. Yeah, so that would be very unlikely, actually, because then you'd find that the minimum one, unless you have like 2 cells that are quite close together, because if you change that, like in the previous example, it's much easier to find one that is the minimum spanning tree.
There were not 7 edge, but 10, or 9 and 9, for example. How would you choose between those alternatives?
I don't think they, yeah, maybe you'll choose the first one, I don't know, yeah. But theoretically, if you have many points, the likelihood of that happening is very, very low, because the points are not that like 9, 9, it will be 9.0, 0.11, 2, 3. So at the end, you will always find a minimum tree if you have many different points.
It's also possible to bootstrap subsample and produce different minimum spanning tree and build a consensus minimum spanning tree.
Yeah.
So
Yeah. Yeah, that's a good point.
Yes.
Yes?
So in sense of gene expression, so what is the minimum? It means that you have a set of genes and another set of genes which is like more close to each other. And
Is it?
This is the minimum distance.
Yeah, so if you consider like these cells here, they are close in the dimensionality reduction space. That means that the gene, all those, let's say 1,000 genes are very similar to each other, and their distance here will be very close. Yeah. So you want to find the closest neighbor of this cell to that one, but not only at the local site, but other, for example, like considering that you have to connect every single point, and the distance between all the points should be very minimal. Yes. Yes. Yeah, most algorithms will try to set a default for you. In my experience, they fail quite often. So it's a 50% chance of getting right and 50% chance of not getting right. So in all those algorithms, there is any at least one parameter that is set up. This is where I should start. Yeah. Yes. Okay. Yes. Yes. So no more questions about minimum spanning tree. So we can go to the next method, which is also implemented in Monocle 2 and many other modern trajectory inference algorithms. So here is what is called reverse graph embedding, RGE. So one of the examples is quite common is DDR3, and there are simple principal trees or principal graph. There are many names for it, and algorithms as well. So the motivation for setting a trajectory using this method is that if you have a minimum spanning tree, let's say you consider these points, and you move one of those points a little bit further, like this one, a little bit further, that completely changes your trajectory. So you're using minimum spanning trees is really dependent on single points. So if you don't have all or many transitional points, that can completely change a minimum spanning tree. So to overcome this issue, what they can do is that try to cluster your samples or your cells beforehand, and then draw a trajectory based on the average of those cells, of those clusters. So ideally, if you have many cells, so then you partition them in subclusters, and then try to draw a trajectory over that. This is what is implemented in Monocle 2, for example, and that's also one of the exercises you'll have. So imagine that you have your points in a multi-dimensional space, and then you do any dimension. So Diffusion Maps, ICA, TSNE, UMAP, you choose. Then you can guess some initial trajectory, design some clusters. So for example, for these cells, a cluster will be here. For these cells will be kind of here, here, and so on. So you can cluster them in different ways and methods as well. So initially it was k-means, as you also heard in the clustering technique. Nowadays, there are many variants. We can do graph clustering, for example, on the other hand. So then, since your cells are quite far from this point, what you try to do is assign those points to the closest part on the tree. So for example, these points would be closer here and that. And then you update the centroids to match these distances as well. And then from this one, you try to reconstruct your data again. So like from a linear to a three-dimensional, and try to compare to this one. And then if it doesn't match very well, you need to reiterate over this several times until what you have tried to reconstruct matches your original data, which is kind of similar what I've talked about when mentioning TSNE or UMAP. So you try to iterate over this so that the dimensionality reduction method or the embedding space closely reflects what you have in the original data. So after you have selected a branching that closely reflects your original data, you just set a root point and then define the distance from the root as being your pseudotime or your trajectory line. And then these also branches can define the cell fate. So some transcription paths can define to one or to another lineage. As I was mentioning, there are many other algorithms. So here's also the most, I think the most newer ones. So this is from this year. So 2018, and many of them are basically doing exactly the same thing as the DDR3, or in some different ways, changing. For example, here they are not using UMAP, neither TSNE. They're using another graph embedding technique, and then you just assign your trajectory and clusters basically as well. Slingshot will do similar strategy, cluster on, I think it's on TSNE. Tscan will cluster on PCA. Router will also cluster on TSNE or UMAP. So there are different flavors of some similar algorithms out there. So pick one and go for it. Yes. UMAP version 3 also implements now these kind of strategy as well. So you have your high dimensional data, pre-processed normalization, PCA that now you're super tired of hearing. Like, stop, Paulo, I want to do something else. So you can reduce to TSNE and UMAP, do some clustering, partition yourselves using this clustering, and then assign a tree. So DDR3, simple. These are all the methods of creating your tree. define the pseudotime based on a branch, and then go for a differential expression. So in most cases, what you have is that you need to define where you start the trajectory, as you have asked before. So where should I start this? So most methods you need to define something, except if you do this new one, which is RNA velocity. There are still some problems, or it's also quite hard to work with RNA velocity, as well as working with the other ones. There are a lot of problems also, I would say. But the underlying method behind it is that is based on a biological concept. That is, the transcription of our mRNA, it first is transcribed as an unspliced version of it. So then you have some introns over here. And after splicing, you have the spliced version of the mRNA that codes for the protein that you're interested in. And then you have also some degradation factor. So what they try to do is to model this as a function of time. So then you can define the velocity of unspliced to spliced version of that RNA molecule. But how does this see in a biological system? So imagine that you have a pseudotime development here. And here you have expression of some genes. They have done many genes in the paper, but here is just an example. So the splice variant, for example, here is decreasing before the spliced version is decreasing at the same rate. The same thing goes for here. They start increasing the unspliced first, and then the spliced ones. So each dot here represents a cell, meaning that the spliced version of that cell is a little bit ahead in pseudotime compared to the spliced version, to the unspliced version. The same thing here. So the unspliced start expressing before the spliced version, and so on. So here you can also see in a different way. So you have these genes, and you have these ones that have a lot of the unspliced version of these cells here. The same thing for this one. In blue, you have the spliced version of these ones here. So then you can now biologically, based on this concept, so that you have this assumption that this is completely true, of course, and that these cells should go to that stage, not to backwards here. So you have now where to set a trajectory. And this helps a lot if you have a big and complex system. So here you have many cells in, I think, large, where is large? It's neuro development, right? Exactly. So that you can define some arrows where the unspliced to the spliced version of that cell is, and you can just set arrows to all of this. So then you can see, for example, that the progenitors of this are somewhere in here, where the cells do not point to anywhere, because the spliced and unspliced version of that cell is still there. So it's kind of a cyclic. Yes?
If you have 3' RNA, come back from the 10x, how much is it actually possible to define or to measure unspliced variants? I mean, the information that we have is relatively short.
You can detect typically 10 to 15% of transcript with intron in 3' and X.
Yeah. So in the paper, actually, there's a figure that I didn't show it here. They compare smart-seq, the cell-seq, I think, and the 10x as well. And they all have the same rates, let's say. I don't know if there is much validation on every single different trajectory, but for this one, it seems to work. Yes? So 10x. Okay, yeah. Yeah. Yeah.
On the 10x web summary, you have the fraction of reads that align to exon, intron, and so on. You can see that actually you have a certain amount of reads that will align to this part, so it's sufficient to infer velocity.
Yeah. Yes?
So, you're saying that if you do convince the pseudo aligners, you want to do exactly the
So you can, with pseudo alignment quasi mapping, actually you just quantify transcript, but you have no idea where the reads actually map. So you can only do that with regular star design.
Yeah.
You need a BAM file actually to produce.
Yes. Yes.
Yeah, the reason why it works in inference, it's thought that there is internal prime. So if there's like a lot of A's in the interval, it binds to the codec key binding on the beat. So there's a sort of internal prime. So that's why you get reads from somewhere in the middle of the transcript, and not only from the pre-prime. Slightly, yeah, but they're long. So, yeah, but yeah.
And because of these directional of the vectors of velocity and transition between one cell state to another, you can actually infer where in your data set, where should be the root and where should be the diffusion points or the ends of your trajectory. So now it's less biased in that sense that you don't need to specify where your biological system should start. And here's just an enlargement of that. So you can see that they start with gaining velocity, so these vectors, until they reach a point in here and disappear. Yes, until the other one as well reaches a top velocity in the end there. And here is also, you can just visualize some other genes as well in one of the branches that is not expressed anywhere else other than this one. So RNA velocity allows a biological driven, or at least model driven trajectory inference. And it also defines the start, the ends, and bifurcations automatically because of this feature. So rather than comparing only the spliced version as all the other methods, you also have now the new information about unspliced, and you combine those two informations to infer trajectories. And the arrows then indicates the direction and the velocity, or how big the arrow is, from transition to unspliced to the spliced version of that cell. Oops. And that is really good because also the RNA velocity can define cyclic trajectories really well. So if you start 1 hour stimulus, you see several cell starts at different points here to get activated. After 4 hours stimuli, they start getting back, but they go back to the original one, which is like beautiful in the paper. So a wrap up. Final considerations. In reality, distance in the multidimensional space also reflects somewhat the difference in transcriptional landscape, but not actual time. So that's why we call it pseudo time. So we need keep that in mind, that we need to have a continuum of states among your cells. If you have two completely separate clusters, that will give you wrong results in the end, or at least the trajectory will not be good enough, maybe. Yeah. And it also may work if you have a single time point collection. So for example, you have a 10x from the bone marrow development, or stem cells from the intestine. But it would be better to have multiple data sets to combine them and have all the chance to get all the transitional cells between the two points. And again, be aware, any data set can be forced to trajectory. And first, make sure that dimensionality reduction and your gene set captures what you expect to. Be aware. So which method should I use? So there is this nice paper that you have the reference here from this year. So do you expect multiple disjoint trajectories? Yes or no? So then you can define, maybe use these methods that use this graph, or you can go for a particular topology. Maybe I know that there's only a bifurcation. Maybe on B cell development, I know it's linear, so then I can go for methods that allow these linear trajectories. If I don't expect, then you can go to, is there cycles in trajectory? Then you go for this one. If no, then you can go for other methods. But in general, most of the ones that define graph trees, like one of those here, they will also define those ones here. And that can be seen in this table from the same paper. So here, they sort the methods based on graph or tree. There are others below as well, but it's too many to show. And here, which trajectory types that you can infer from it. And RNA velocity was not included because it was published kind of almost together. But it's discussed in the paper as well. So here I have Monocle ICA, Monocle DDT. Now you know what MST is. PAGA is kind of also based on dimensionality reduction and clustering and tree building.
nice to see we have LP graphs that has been developed in Institut Curie.
Yeah.
In the middle of nowhere.
This one?
Just LP graph.
This one. Okay, yeah. Here. Then you have, if you really don't like R, you want to Python, you have also Python versions in here. You can, but yeah. So it's a quite good review of overall the methods. But really, if you choose any of those 3 methods for these cyclic trajectories, it will be fine. If you have 3 building ones, also most of them also will provide you with kind of similar results. Yeah, and that's it. So